Ji, Assalamu alaikum everyone. How are you, second year? Today we will discuss the endocrine secretion of pancreas, focusing specifically on the molecular structure and biochemical function of insulin. Uh, by the end of this session, you will be able to list the various pancreatic hormones, explain the synthesis and chemical structure of insulin, and you can also describe the mechanism of action, metabolic functions, and regulation of insulin. So let's begin. Let's define our primary focus. Insulin is a peptide hormone secreted by beta cells in the pancreatic islets of Langerhans. Um, its primary physiological role is twofold. It lowers serum glucose level um, and promotes the anabolism, the storage of energy. Beyond metabolism, insulin is also essential growth factor required for normal human development. Looking at the macro to micro structure of pancreas, we see an organ with dual roles. Pancreas, we see an organ with a dual role, okay? And there's exocrine and endocrine role. With the bulk of the pancreas is exocrine. The endocrine is housed in scattered microorgans called islets of Langerhans, and which makes up to 1 to 2% of the total pancreas mass. And within these islets, we find a cellular matrix where alpha cells and delta cells, beta cells, F cells, um, ये सब सारे cells होते हैं. And alpha cells secrete glucagon, beta cells produce insulin, the delta cells secrete somatostatin, and F cells secrete pancreatic polypeptide. Okay? And these exocrine cells, they are acinar cells and ductal cells. So this is the whole table where you can see that alpha cells secrete glucagon, beta cells insulin, delta cells somatostatin, and pancreatic polypeptide cells, they secrete pancreatic polypeptide. This diagram illustrates a function split. The endocrine release hormones like insulin and pancreatic polypeptide, glucagon, somatostatin, and in contrast, the exocrine or digestive enzymes uses the acinar cells to produce enzymes like lipase, protease, amylase, okay, and which are secreted through ducts into small intestine to aid digestion. Then we have chemical structure of insulin. Now let's look at the molecule itself. Human insulin is 51 amino acid peptide with a molecular weight of approximately 5.7 kilodalton, and it consists of two polypeptide chains. Okay, these are two polypeptide chains. This is A chain, this is B chain, and um, this chain has 21 amino acid residues, and the B chain has 30 amino acid residues, and these chains are held together by two interchain disulfide bridges connecting A7 to B7. This is A7, or ये connect करिए to the B7. ठीक है, this is the B7, and uh, A20 to B19. Okay, A20 to B19, and there is also a critical third interchain disulfide bridge within HN itself, okay, connecting 6 to 11th amino acid residues. All right. So this is the overview of the biosynthesis of insulin. We have three major steps in it, and it begins with the synthesis of a large precursor um, called pre-proinsulin, which is then converted into proinsulin, and which is then converted into insulin. ठीक है. So insulin is synthesized by ribosomes of the rough endoplasmic reticulum as a large precursor polypeptide that is then converted to mature hormone prior to secretion. And how it is synthesized we will see in our next slides. So this is the synthetic pathway, and this is the whole assembly line, and we start with the pre-pro insulin, which contains a signal sequence. Okay? Then it enters the endoplasmic reticulum. That signal sequence is cleaved to form proinsulin. See, here we have amino end, and this is the carboxylic end of the amino acids and protein. Okay? And this is the signal sequence. And when it enters into the endoplasmic reticulum, it leaves this amino part, and it is now left with the proinsulin, which has got uh, this B chain. Okay? And this is the carboxy terminal, and this is the chain of the C peptide, which links this B chain to the carboxylic terminal. Okay? And then when it enters the Golgi apparatus finally and secretory vesicle, the C peptide is also removed, leaving us with the active A chain and B chain that constitutes the mature insulin. So this all A chain and B chain, this makes the mature insulin, and it is left with only a C peptide. All right. This figure is showing the translation occurs at the ribosomes, okay, where the signal sequence directs the chain into endoplasmic reticulum, and inside the lumen enzyme chopped off the signal sequence and disulfide bomb to form a stable proinsulin structure. See, this is the pre-proinsulin, and when ribosome translate into precursor chain, it includes a signal sequence that directs the chain into endoplasmic reticulum, and this is how the proinsulin is formed. And the enzyme chopped off the signal sequence, disulfide bonds form to stabilize the structure, and this is where when it enters Golgi to secretory vesicles. This is the mature natural insulin, and proinsulin enters the budding vesicles. Enzyme cleave the molecule into active fragments packed into the granules, awaits release. Okay, so this is the mature insulin, and here you can see the A chain and B chain, and C peptide is also there. All right. Here you can see the storage versus action, the zinc vault. So within the B cells, insulin is stored in a stable hexameric form, and this hexamer is organized around two zinc axial atoms, creating a robust structure that prevents degradation. Um, However, to be biologically active and binds to receptor in the target tissue, this hexamer must dissociate into its monomeric form. All right. So this is the monomeric form, and this is the biological active form. And यहाँ से जो hexameric structure है इससे निकल के this forms the active form. This is the mechanism of action. When insulin reach a target cell, see, insulin reaches target cell, it interacts with the specific membrane receptor, and it triggers a signal cascade that causes vesicle containing glucose transporters GLUT4. ठीक है, GLUT4 इस तरह आता है, and then it moves and fuses with the plasma membrane and increases the number of the transporters on the cell surface, allowing glucose to enter the cell. And when insulin level drops, these transporters are removed from the membrane via endocytosis and stored back in the vesicles, ready for the next cycle. So this is the first step, and this is the second step, third step, and this is the smaller vesicular form with large endosome, and the patches of the endosome enriched with the glucose transporter buds off to become small vesicle ready to return to the surface when insulin level rises again. So let's look at how the beta cell decide to release insulin. It's a beautiful domino effect. And first, glucose enter the cells. Okay, this is the step one, where the glucose enter the cell through GLUT2 transporter when blood levels exceed 3.9 millimoles per liter, and the glucose is then metabolized, increasing the ATP to ADP. Okay, and this spike in ATP causes the ATP gated potassium channels to the date will be closed, and ATP potassium channel closed, stopping potassium efflux. Then there is depolarization, and the trapped potassium depolarize the cell membrane, and then there is a calcium influx, and where depolarization forces the voltage gated calcium channel to open, and the calcium floods inside into the cell, and then um, here you can see the vesicles, um, the insulin packed granules to fuse with the membrane, release their contents into the blood via exocytosis. So this is the whole domino effect of the secretion of insulin. Now you can see the tyrosine kinase and translocation. So once the insulin reaches its target tissue, it must knock on the door, and it binds to the alpha subunits of its receptor on the cell surface, and activates the tyrosine kinase. Activity of the internal beta subunit initiate a signal cascade, and this signals tells this cell to move GLUT4 um, storage vesicle to plasma membrane. Once fused, these transporter allow glucose to flood into the cell, and this is how insulin directly lowers blood sugar by moving into the muscle and the fat tissue. See the receptor and dynamics downregulation, and the cells also has a way to manage its sensitivity after insulin binds. Uh, the entire hormone receptor complex is often internalized and or envelop into the cell, and most receptors are recycled back to the surface, but the insulin itself is degraded in the lysosomes. And crucially, if the insulin levels stay higher for too long, as seen in chronic hyperinsulinemia, the cells may degrade the receptor rather than recycle them, and this downregulation bottleneck is a fundamental cellular mechanism behind the target tissue insulin resistance. And on this slide, you can see the insulin, the regulator of opposing forces of the secretion. And the insulin secretion is a balanced act. And major stimulators include nutrient drivers like high serum glucose, amino acids, fat-free fatty acids, and we also see GIT hormones like GIP and gastrin, and conversely, um, uh, uh, they provide a feed forward signal all after a meal, and we see also inhibitors including nutrient deficit like fasting or low glucose levels, and hormone suppression from somatostatin, and the autonomic nervous system can also override these signals like epinephrine through alpha 2 receptors, and can inhibit the secretion during stress. uh, um, the transport network, that GLUT family metrics, and these uh, not all glucose transporters are the same, and GLUT1 and GLUT3 provide basal uptake for most tissues at a steady rate, and GLUT2 found in the liver and beta cells, and has a lower affinity, that is high AKM, and allowing it to act as a glucose sensor that only reacts with sugar when sugar is high. And GLUT4 is the one we can care about for insulin therapy, and it is insulin dependent and found in muscles and fat cells. And finally, GLUT5. Uh, GLUT5 is unique because it's primarily it transports fructose in the small intestine. Then we have secondary physiological actions, like insulin does more than just manage sugar, and it's a critical regulator of potassium homeostasis and actively driving potassium from extracellular fluid into the cell. Okay, and clinically, this is why insulin is sometimes used to treat life-threatening high potassium levels. And additionally, insulin promotes vascular health by stimulating the expression of endothelial nitric oxide synthase and release nitric oxide to help blood vessels dilate. So thinking of insulin as the master switch for anabolism storage, and in the liver it turns on glycolysis and glycogen synthesis while shutting down glucose production, and the muscle drives glucose uptake and protein synthesis while stopping protein breakdown. In adipose tissue, it promotes the fat storage by activating the lipoprotein lipase and inhibiting hormone sensitive lipase to prevent the breakdown of stored fats. And hmm, this is the summary, the ledger of the insulin, and stimulates the entry of the glucose into the muscle and glucose tissue, the synthesis of glycogen, and the synthesis of fatty acid and proteins. And at the same time, the body primarily inhibiting the breakdown of glycogen, the production of new glucose in the liver, and the metabolization of fatty acid from your fat stores. Then finally, let's look at the vital statistics and see the this insulin produced by the beta cell as a 51 amino acid peptide, and it has a short half-life of about 5 minutes, allowing for rapid systemic clearance and precise control. It dissolves freely in the plasma, targets a membrane receptor with tyrosine kinase activity. And remember, while it controls the whole body, the brain, kidney, and intestines are not insulin dependent for their glucose uptake. So this is the temporal action timeline, and you can see here that um insulin response, the fed state, and the uh, this is the short-term effect, second to minutes, and you can see the immediate receptor activation and activation on the transport system and covalent modification, that is phosphorylation and dephosphorylation of pre-existing enzymes. This is the short-term effect, and this is the intermediate effect where induction of specific gene is there and expression of newly translated protein is there. And then the long-term effect, that is from hours to days, that stimulation of DNA synthesis and cell proliferation and differentiation. So these effects must result from simple linear pathway, but from multiple diverging and converging pathways mediated by the insulin receptors. So this is the fantastic technical diagrams to include and in our lecture. And this is a technical discussion, and let's look at how we actually produce the insulin used in the clinical practice today. And before 1980s, we relied on bovine or porcine insulin, but thanks to the recombinant DNA technology, we can now manufacture identical human insulin using E. coli bacteria. So there is no more bovine or porcine insulin used now as medicine for diabetes. So the preparation and the process begins with isolating the human gene that code for A and B chains of insulin, and separately we take a bacterial plasmid and use restriction enzyme to cut it open. And when they inserted the human insulin gene into this plasmid, creating that we call recombinant DNA, and these recombinant plasmids are inserted back into the E. coli, and these bacteria act as macroscopic factories. We place them into the large fermentation tanks, and where they replicate rapidly. As the bacteria grow and multiply, they faithfully follow the instructions in the recombinant DNA to synthesize large quantity of human insulin A and B chains. And once fermentation is complete, we extract and purify the A and B chains from the bacterial broth, and but they are still we can separate them. And in the final chemical step, we bring the two chains together and use the formation of those critical disulfide bond and that we discussed earlier in the lecture. And this ensures the molecule folds into its proper biological active 3D shape. So as a result is humulin. Most of you must have come across this name. This is the synthetic human insulin, and this technology is the landmark in the medical history because it provides a pure, non-allergic supply of insulin that is chemically identical to what is produced in the human pancreas. And it's a perfect example of how the biochemistry you learn in the lab translates directly into the life-saving treatment at the bedside. Here in this slide, you can see that how the extraction is being done. Human cells DNA is isolated, and then specific gene for the insulin is extracted using restriction enzyme, and then plasmid isolation. There's recombination, there transgenesis, and then there's cultivation, and there is harvesting, leading to the formation of the synthetic insulin, which we use for the diabetic patients. All right, so that's all, and I hope it makes sense. And if you have any queries, you can contact me. Okay, thank you.